Hi everyone, I'm Carlo Pettirossi and I'd like to welcome you to an interview offered by Atomi per la Pace. This is actually our first interview and I'm particularly proud to introduce on behalf of all our staff our exceptional guest, Dr. Edward Calabrese, Professor of Toxicology at the University of Massachusetts. Professor Calabrese, thanks very much indeed for being with us.
Thank you very much, appreciate it.
Before letting you introduce yourself and your work, I'd like to give a very brief introduction to the viewers. Atomi per la Pace knew about part of your work thanks to a junk science article found on Rod Adams' blog Atomic Insights, the link of which can be found in the description box to this video. By the way, Rod Adams wrote several other articles about your work, the latest of which is dated March 28. It talks about the presentation you gave at the Cato Institute on March 21. Of course, the viewers will find the link to this article in the description box too. This junk science article, which contains links to two of your papers, both issued in 2011, talks about the LNT model, which is used by every nuclear regulatory commission worldwide as a radiation dose response model to follow in order to determine the upper limits within which the human body can be exposed to radiations during a certain period of time without permanent consequences. Well, according to your studies, this model is nothing else but the result of a scientific fraud. It gets even worse than this. In fact, the main character in this fraud, Hermann Muller, got a Nobel Prize for it. Well, I guess it's time now to leave our guest to the talking. Professor Calabrese, please give us an introduction of yourself and of your area of expertise.
My name is Edward Calabrese, and I'm a professor of toxicology at the University of Massachusetts in the School of Public Health in the Environmental Health Sciences Division. And my area of expertise is toxicology, in which I'm board certified. I've been at the University of Massachusetts since 1976. I'm a traditional faculty member where I teach 3 courses per year, and I have an active research program. And I have been very active in the area of dose response since approximately 1985. So for, basically well over 2 decades now, going on 3 decades. And prior to that, I spent probably about 10 or 12 years with a particular focus on studying inter-individual variation in susceptibility to pollutant toxicity and carcinogenicity, trying to find out why some people get sick and others don't. And that led me to looking at animal models that would be predictive of high-risk groups and things of that nature. And eventually, all roads led to a better understanding of the dose-response relationship.
I'd like to invite the viewer to consult your page on the UMass site for further details about you. First question, can you please tell us how and why you came up with the idea of criticizing the author of the LNT and his model?
Well, it happened by accident. It really happened as a result of the fact that I had written a manuscript that I was submitting for publication that was concerning historical development of the LNT. And I was going to submit it to a leading toxicology journal. And in the course of preparing this, a standard mode of operation for me is that when I get to a certain point in the manuscript's development, I will oftentimes send it out to a number of people that I'd call friendly critics who would evaluate it and give me their honest criticism before I would submit it for publication consideration to a journal. And in the course of this, I sent it to a colleague who is extremely well known in the area of toxicology. And I sent it to other people, but this particular person came back with a criticism that was specific but somewhat general. And he indicated to me that that I needed to address the role of Dr. Herman Muller better, more insightfully in the area of the history of the dose response, especially the linear dose response, and that draft of my manuscript was not sufficiently informative, just did not go into depth. And so what happened was that I agreed with this criticism and realized that even though I had read some about Muller and I read a biography concerning Muller, I really hadn't dug out his life well enough. And so what that did was it forced me to get many of the papers that Muller wrote, which were a considerable amount in his career, starting with the very first and taking it up to when he died. And then also rereading the biography, also going back and rereading and studying his Nobel Prize lecture. And then that led me into wanting to know more about Muller and the intimate details of his involvement with other leading radiation geneticists of the time. And so what happened is that I was interested and I was able to get a purchase from the University of Indiana, where Muller's papers are held. Numerous, really hundreds and hundreds of letters that he wrote or other types of correspondence or documents. And then I went to the American Philosophical Association in New York City. I think it's New York City or Philadelphia, I think it is. And purchased other kinds of communication that other people had with him that weren't contained in his own files, so that I got a very substantial quantity of information. And in the course of this, I learned a lot about Muller, and I was really trying to understand how the field came to switch from a threshold model that was very dominant up until the mid-1950s, how it came to switch to a linear model for cancer risk assessment. And actually what happened was that I have learned that Muller was very strongly advocating linearity during the 1930s and 1940s, and certainly 1950s, his whole life, really. But he wasn't having a lot of success in convincing the regulatory community and governmental agencies to adopt his views, even though other people within his field, which would be radiation genetics, I think strongly in general with his view of a linear dose-response relationship. So in going forth on this, the studies really were not particularly convincing or clarifying going into World War II. And during World War II, the U.S. created a program called the Manhattan Project, which was really to lead to the production of the atomic bomb. But in doing that, the U.S. government had many different aspects of the Manhattan Project. And one of them was to better understand the nature of the dose response in the low dose zone for essentially radioactive, for X-rays and for gamma rays and the radionuclides. And so what happened is that the U.S. government, the Atomic Energy Commission, gave a grant to the University of Rochester, and one of the recipients was a well-known geneticist, Stern. And Stern was to conduct key studies on dose response using the fruit fly model and to try to answer the question of the nature of the dose response in the low dose zone. And as it turns out, Muller was at that time a professor at Amherst College in the town that I have been living in for the last 37 years or so, living 1 mile away from where I'm sitting today. And Muller was retained by the Manhattan Project at the University of Rochester through the actions of Kurt Stern to be a consultant to that project. And so Muller provided lots of information. He provided one of a strain of fruit fly. He was very helpful in telling them what kind of study design they should have. He was extremely important in attempting to resolve questions on control group, a spontaneous mutation rate. He reviewed manuscripts for publication. Muller was very involved in what this research activity was. Now, basically what I learned, and I'm sorry for the long story, but it leads to an answer here, and that is that in the course of this research, what happened was that two large studies were to be done. One was of an acute exposure to the fruit flies, an exposure that would be given to X-rays in a very, very short period of time. And then in that particular study that was done with Kurt Stern and Warren Spencer, and Spencer was a well-known Drosophila geneticist, they showed that there appeared to be a linear dose-response relationship. However, the real test of this dose-response question was to be resolved with a chronic exposure study. And Ernst Caspary, another well-known researcher, was working with Kurt Stern on the team, and his job was to do the chronic study. The chronic study would result in exposing the fruit flies to a dose rate. It was essentially about 1/13,000th of the dose rate that was administered by Warren Spencer in the acute study. So it was a very, very different kind of study. And it also was a study that incorporated a number of methodological improvements over what was done by Spencer. Now, as it turns out, when Caspary got his findings, he went and shared those with his superior, which was Kurt Stern. And Caspary's data did not support a linear dose-response relationship. It supported what appeared to be a threshold dose-response. Well, what happened at that point was that Stern did not want to accept Caspary's findings and challenged him. He claimed that Caspary's control group was aberrantly high, and that was the reason why the findings led to a threshold interpretation rather than a linear interpretation. Caspary decided to dig into the literature and found a number of studies which did not support the position of his superior, Kurt Stern, but that supported the reliability of his own controls. And so then Kurt Stern contacted Dr. Muller in Amherst and said, can you share with us your data, because you have been looking at the question of spontaneous control group mutation in the same model that we've been using. And so what happened was that he provided copious amounts of data to Stern and the others, and as it turns out, Muller's data supported the interpretation of Caspary. And so what happened is that in letters that I have between Caspary and Stern, and also between Muller and Stern, I have them basically, I have Stern backing down, reasserting, in fact, the interpretation, the original interpretation of Caspary, and in fact, where the data supported a threshold. Now, what happened is that when I was going through this, as it turns out, I wasn't, I've jumped ahead in the story a little bit. See, at that point, I wasn't sure just how much Muller really knew of the conversations between Caspary and Stern. I was to learn later how much he knew. However, what I was wondering about was, I said, wow, Caspary showed in this very excellent study that the data supported a threshold. And that this was the strongest study by far that had been conducted at that point. And then I also recalled, now this was in August, September, October of 1946. Now, in December 12th of 1946, Dr. Muller receives the Nobel Prize and gives a speech over in Stockholm accepting the Nobel Prize. Now, if you read his speech, what he says in this is essentially relates to the dose response. He says that one can no longer accept the use of a threshold model, that the threshold model needs to be replaced by a linear model, and he really lays this out in very strong and definitive terms that threshold is really, intellectually and scientifically dead, and it needs to be replaced by this linear model. And so I said to myself, having read this and studied his comments, I said, I know he was consultant to the Manhattan Project. I know he communicated with Stern and the investigators. I said, but had he really read Caspari's manuscript that showed the data, really showed the data, and it supported actually a threshold. In fact, Caspari was advocating for a tolerance dose or threshold in the manuscript. And I said, well, I've consulted on a lot of studies in the past, and sometimes the people share with you information, and sometimes they don't show everything. So I didn't really know what he knew, how much he knew. I said, did he really see the paper before he gave his Nobel Prize speech? And so I had some doubt. But in the course of going through all the letters that were sent back and forth between Mueller and Stern and others, lo and behold, I found out that on November 6th of 1946, Stern sent Caspari's manuscript to Mueller asking him to review it. Mueller receives it and writes a letter back to Stern on November 12th, saying, I received the manuscript of Caspari. I've pretty much skimmed it over, went over it quickly. I can see that this is a really serious challenge to the linear dose response model. The study needs to be replicated. I don't have any reason to doubt the capability of Caspari to do a proper study. I will get back to you with detailed comments before I take my trip to Europe. And so at that point, as far as I was concerned, he was aware of it. He knew what the implications were. He had some sense as to the credibility of the people who did it. Of course, he was a consultant for them. He was advisory on a lot of what they were doing. But I was still waiting for his major review. So then he goes to, when he goes to Stockholm, at Stockholm, as I mentioned before, he basically goes headlong the opposite direction, that there's no possibility of a threshold. None whatsoever. You have to change. And yet he had actually seen in front of him that there was a very strong challenge to a threshold. And he said it had to be replicated. Now, replication of these kinds of studies is not trivial. It involves at least a year, maybe more, lots of money, multiple sets of expertise, and you don't want to waste anybody's time, money, and everything if it's not significant. And so it was significant enough for him to say that. And as it turns out, what he should have said, in my opinion, was that there's still some uncertainty. We have to do more research to resolve the question of the dose-response relationship. Well, there wasn't enough data out there to say you can no longer consider the possibility of a threshold. He basically, in my opinion, was misleading the audience. Now, people could say to me, well, maybe he changed his mind between November 12th and December 12th. And so, but what I did was I also was lucky enough to get a January 14th, 1947 letter, 6 or 7 page, singly spaced letter from Mueller to Stern, in which he gave his detailed evaluation of the first Stern results. And in it, it's almost an opening statement. There's really almost nothing I can add to this. It's a very well-conducted paper. He reasserted that the linearity was challenged, that the paper, the research was so significant it needed to be replicated as soon as possible. He didn't necessarily believe at all that the threshold was the correct interpretation, but that's why you do the additional studies. The studies had to be done to resolve this question. He didn't have any technical issues, and basically the issue was, let's get this replicated. And so as far as I was concerned, what happened was that Muller's opinion had not changed. Muller reaffirmed his original opinion, and it just raised even further questions to me that how could he have made his statement to the Nobel Prize Committee, knowing what he knew and his opinions didn't change. It's like in private he was revealing of his true feelings to Stern, but in public he was giving a different story. He was really, in my opinion, misleading, deceiving, and being dishonest to the public. While in private he was being, a good scientist. And you really can't have it both ways. And so essentially what happened is that, so I didn't go into criticizing Muller because I had any axe to grind with Muller, that I had any issues with him, that I had an historical problem with him. I actually ended up criticizing Muller more or less as an unexpected byproduct of trying to produce a better paper. So sorry for the long story, but that's how I uncovered, so to speak, the beginnings of this controversy over how we transformed, from a threshold to an LNT. And I think it really started with this major act of deception, this major public display of scientific aggrandizement, and where we put all our major achievers, basically, on display, and try to listen and learn from them, and you hope and expect that they are telling you the truth. And he wasn't.
Okay, next question. Can you give us some more details about Muller's and Casperi experiments?
Yes, I can tell you about it. As I alluded to, there were two experiments done. There was one with acute study, and there was one with the chronic study. And I think this is important, and it'll get back into your question. And that is that you see, the reason why Kurt Stern challenged Casperi is that he and his radiation geneticist community really believe very strongly, or wanted to believe very strongly, in a linear dose-response relationship. And Casperi's data did not support it. And so, first there was the challenge that your control group was wrong. And now, so Casperi answers that question. But a very interesting thing here, and that is that if you read the Casperi paper that was published, co-authored with Kurt Stern on this topic, which was published finally in 1948, almost the entire 6-page discussion was a disclaimer of, even though our data is what it is, please don't accept and use our data until you or we can explain why our findings differed from those of the earlier acute study done with Stern and Warren Spencer. Now, it's interesting that Kurt Stern forced the chronic study to try to explain why it tipped from the acute, but not the other way around. You could believe the Spencer data, but you couldn't believe the Casperi data. That was actually interesting right there. But beyond that, I identified about 25 differences, methodological differences, between the acute study and the chronic study. There were differences, first of all, in the acute study they used X-rays, and in the chronic study they used gamma rays. In the acute study, they gave a direct exposure to the males. In the chronic study, they actually, the sperm copulation had taken place, and the exposure actually was given to the sperm as they were stored in the female. So different, the organism was actually different that was actually exposed. The diets were totally different. The females were put on in the Casperi study on a diet that would in fact prevent the laying of eggs, whereas it was a totally different diet in the other case. There were, as I said, 25 differences between the studies at least, such that you actually couldn't go back and ever figure out exactly why one study would have differed from the other, because you had too many differences between them to try to, resolve 25 simultaneously differing circumstances. Usually in experiments, you keep everything constant except for one variable. In this case, there were 25 differences between the studies, and yet, Kurt Stern, he's a very significant gentleman. He's got lots of experience. Muller's got as much experience, if not more, than Stern. And Caspary, he's very talented himself. They all knew that you could not go back in and resolve these differences. But nonetheless, in this paper, that's what they're telling the reader to do. And so it made no sense to me, even looking back so many years, that these people who are really outstanding individuals, great intellects, really all of them, that they could have written this and that anybody else could have believed them. Now, the interesting thing was that after Muller read the paper, Muller's name was added to the paper as a consultant, and the only other change that happened in that manuscript, this was the impact of Muller. The only change was that Caspary and Stern removed any reference to this being a threshold phenomenon, and so that was basically, it was only like a minor change in a sentence, but it removed the key word of a tolerance threshold dose response. And so the interesting thing that happens along these lines, in my opinion, and that is that that at some point in time, this is going to be, it's going to be revealed that in fact the Caspary data are going to be published. It's going to support a threshold, and Muller has just said to the world's audience that there's no possibility that a threshold could ever exist. So, and basically, and him having seen the Caspary data, having been a consultant to it, that this has a possibility of basically challenging his credibility as a scientist and as a Nobel Prize winner. And so what happened, several things happened now, and that is that Muller's recommendation that in fact the Caspary study be replicated actually took place. It took place at the University of Rochester, and Caspary was leaving, going for a new job, and Spencer was going back to his old job. So they needed a new person to take over and do the actual work. So they got a young lady who was a new graduate student by the name of Delta Uphoff, and she came in to work with Kurt Stern to make this happen. And so she replicated some of Caspary's work. In her first experiment, she came in and shared her data with Stern, and her control data was reading significantly below that which was expected. It was reading about 40% or so below what was reported in the literature, and about 40% or so below what Muller's data had shown. And so what happened was that, in fact, Stern and Delta Uphoff decided that their findings did not have credibility. And they wrote their manuscript up based on this experiment. And in it, they claimed that their data were not interpreted. And they claimed that the problem with their data was that her control group was aberrantly low. And they cited Caspary's work, the literature. They also cited and thanked Herman Muller for submitting his data for them to review, and he was specifically acknowledged as thanking him for allowing them to use his data. So he actually had a positive affirmation that he knew what was going on. And so she went back and did a second experiment, and she got essentially, oh, by the way, before I do that, in the discussion of that manuscript, the writers, and I suspect that this is what Kurt Stern must have been saying, but I've never seen this before, and that is that he said that one reason why they might have had the aberrant findings was in fact due to investigator bias. And so I suspect that he was referring to not himself, but to Delta Uphoff, but that was never clear. But there are only two names on the paper. And so she went and tried to do a second study, and she had the same problem with the control group, another aberrantly low value, and they couldn't use that data either. It was still in the uninterpretable zone. She did a third experiment where the results for the control group came out, which appeared to be normal to me, but the low dose that she was studying at, if you were to take a look at this from the point of view of a linear dose response, the value she got was something like 3 or 4-fold greater than what would have occurred with a linear prediction. And so when one looked at this, it appeared that her low dose response was aberrantly high, kind of comparably to her control groups and the two experiments being aberrantly low. It appeared as though Delta needed more experience in doing these kinds of research, and essentially the findings were, in my opinion anyways, the three experiments didn't have a lot of credibility, but they had really, so what happened is this, that we're in a situation now where Stern decides that essentially he's trying to get this work published, and so he takes the Spencer paper on acute, which he's already published now in his journal Genetics, and he's taken the Kasperi paper, which he's already published in his journal Genetics, and what happens is, in fact, he takes these two findings in the 3 Delta Uphoff experiments, and he rolls them all together in a type of what I call modern-day meta-analysis, and he tries to make sense of them. Except what happens is that he presents this only in a single table in a 1-page paper in the Journal of Science, and what he says is, more or less, is that the 2 control group studies that were aberrantly low, he now says are normal. He now can interpret them, but he doesn't share with the science audience that less than a year before they were uninterpretable, that their results were aberrantly low. He doesn't go back and say, how has the data changed that these now become normal? Because the database had not changed, and any studies that came afterwards actually would have reasserted, in fact, the aberrantly low nature of those control group findings. Now, what happens is that he also goes back into this science paper, and he now claims that, in fact, the findings, the control group and the control group of Kasperi and Kasperi's work, had irregularities and were now not reliable. And so what he did was he backed into his original position before Kasperi had essentially refuted him, now claimed his data weren't reliable. He then took the Delta papers that really were not reliable, that he had in fact, claimed not to be reliable, made them reliable, and then he added them all together and came up with a linear dose response relationship that supported, the LNT, the foundation of LNT. And so he got those in Science, and then he promised all the science readers, the scientific community, that he would follow through with a detailed report on all the methodology, all the data that they couldn't show, where you could actually see variability, methods, strengths, weaknesses, the whole bit. However you worked your experimental system. And he never followed through with this publication. And so what happened is that 2 papers became very significant then in the radiation genetics research community. It was the Warren Spencer work, which showed linearity for the acute exposure. And then it was this meta-analysis that Stern did with Uphoff in Science, in which they had all these sleight of hand changes, making something appear real that 1 year before was not real, that they didn't support, and basically not sharing that with anybody else, Most of this was hidden in for some point in time, the classified literature of the American Atomic Energy Commission that was never broadly available, and it, essentially becomes a story that was to be discovered. And that's what I actually discovered when I went back in and started digging through all this and found the manuscripts that other people hadn't read or seen, the correspondence of this. And the interesting thing here is that there were two things going on. One, this paper for Science was very significant because it really reaffirmed the belief in linearity. In other people of that time started citing this, and they said, essentially, Kurt Stern has worked with 50 million fruit flies. How can 50 million fruit flies be wrong? It was everything points to linearity. The Caspary study had an unusually high control group. You couldn't believe its findings, so therefore that has no credibility. And a lot of things that were wrong or misinformation. It's like day became night, and night became day, and truth became falsehood, and falsehood became truth. And it was, and nobody was trying to follow the P, with the person who was showing you the trick. You had to follow the data. And there was a very high, I believe, appeal to authority within the community, to the people like Kurt Stern, like Muller. Now, part of this was going on that they're really trying to affirm linearity, but they also had to protect Muller's reputation. Muller really had misled the scientific community at the Nobel Prize. And if Muller had his reputation damaged, it would really hurt any arguments for LNT. So both had to be protected at the same time, in my opinion. Now, in 1950, I was interested in how Muller responded to all this in his own writings in the literature. Publishes two significant papers in 1950. And this is really hard to believe. But Muller goes in one of these papers and says, well, this Stern, Caspary published a paper that deviated from Stern, but when we went back and looked at this, its control group was aberrantly high. And so Muller, whose data was used to support the reliability of the Caspary findings, now concludes falsely that his Caspary's data were aberrantly high when Muller actually came to Caspary's defense. And nobody challenges Muller on this. Not even Caspary challenges Muller on it. It was a blatant misrepresentation of the record by a Nobel Prize winner. In the aftermath of his deceptions during the Nobel Prize, because it actually gets even worse. It's like a lie is piled upon another lie, and nobody challenges it. And I have tracked every single communication in letter and cable between Muller and Stern during these time periods, and see how they went through and how they tabulated and however they communicated. And these are contained in manuscripts and for publication. And so, but another thing is that he basically goes in and he tries to take the lowest dose that was done in the Caspary study, and he rounds down the value so that he can extend the range over which he claims linearity occurs. And so even if linearity was true, which the data really did not support it, you could have claimed a range over, let's say, 250,000 range of dose. Whereas with Muller rounding down incorrectly, he extends it to 400,000 fold. He does different things in which he's either wrong or he's dishonest, and other people actually cite him as the authority. And so what happens is that, now this is interesting, because all this is leading towards something. It's Muller's goal, Stern's goal, and their colleagues' goals are to really change the risk assessment paradigm. And it's to have radiation, ionizing radiation, seen not as a threshold phenomenon, but as a linear dose-response phenomenon for risk assessment purposes. And in 1955, the Rockefeller Foundation provided funding to the U.S. National Academy of Sciences to put together a very distinguished, broad-based group called the Biological Effects of Atomic Radiation, the BEAR-1 Committee. It preceded the BEER committees that we currently have now, in the United States National Academy of Sciences. There was a BEAR-1 and a BEAR-2, and then it shifted to BEER-1, which is they just changed atomic to ionizing. And so that's the only difference in it. But the interesting thing is that I knew that there was essentially only that there had to be a battle coming up between those who supported threshold and those who supported linear. But as it turns out, if you get the transcripts of this first ever BEAR-1 committee, which I have obtained, and you read them all from cover to cover, back and forth, inside and out, you find that there is no debate on the nature of the dose response in the low dose zone. It is accepted from the moment they walked in that the dose response was linear. And when you look at the comments of Muller and the comments of other members of that committee and the written language prior to the committee, they claim that in the early 1950s, the decision had been made amongst this radiation genetics group that it was no longer a threshold, that it really was a linear dose response. So there was no debate. It was what happened was that the committee came stacked with essentially a very large proportion of radiation geneticists who are of the same mind. And because they were of the same mind, the decision basically was automatic, and it went right to linearity. And basically, as a result of this, and then when you go back and you look at the literature that they all go back and cite is the Spencer study and the Uphoff and Stern study in Science. These are the two critical references that basically were the ones upon which the switch in that BEAR-1 committee were really pivotal and were based upon. And in my opinion, the key one was the chronic study, and that was the Uphoff and Stern one, and that was really the one, in my opinion, that was pretty fraudulent in ways that were as I described. And so what happened then, within a year after the BEAR-1 came out with their recommendation, the NCRP decided to recommend that the findings that were for germ cell mutation and linearity be generalized to somatic cells, and that just opened up the application to cancer risk assessment. And ever since then, it's just followed a linear dose response relationship. And this not only had its impact on ionizing radiation, but then the US EPA years later essentially took the same rationale and the same basis and in fact applied it to chemical carcinogens and just generalized even further. And that's been the regulatory history for cancer risk assessment in the United States and essentially most other countries throughout the world. It actually is a very terrible history. And it's a foundation that is based upon, misrepresentation and at the highest possible level from the people that you actually are dependent upon.
Are the original letters between Muller, Stern, and Kasperi available somewhere on the internet?
They're not available on the internet. I can certainly send you my copies. They are publicly available in the sense that from the same sources that I got them from. The Mullis papers are at the University of Indiana, the Libby Library, and Stern's communications are at the American Philosophical Association. However, I have published different articles concerning these letters as part of the papers, and it is not uncommon for the editors to require me to show proof of letters. And so I have had to provide, I'd say, scanned copies of the letters to the editors or to the reviewers that they probably share the letters with, because these are very, very specific kinds of information that I'm claiming, and for somebody to actually affirm that I pass a peer review process, I have to provide documentation and proof to the editorial judgments of these independent journals, that and they have to actually have that as backup to protect themselves when, in fact, my work gets criticized. So as part of their own, what you would call due diligence to the peer review process. So, but I could certainly, send you a copy of my letters that I have obtained. But the journals themselves, for the most part, I am required to provide proof. If what I am having is not generally available, and these letters would not be considered generally available because you'd have to go in and purchase them, and so it's not immediately available. That's why I think I have to provide them to the journals.
Why do you think that the Rockefeller Foundation put up the Bear Committee?
I actually don't know the answer to that question. I know that Rockefeller the foundation was a strong leader in many aspects of the biological sciences, had a strong social, political conscience. And why they picked this topic, I suspect that it was 1955. There was a lot of, since I was a child during that time period, there was a lot of tension, Cold War tension in the 1954-55 time period. And I believe, and there was the atmospheric testing of atomic bombs. And I think that they wanted to better understand what the public health risks might be from probably atmospheric testing, but perhaps broader issues with the new developments of nuclear medicine, nuclear energy, lots of new world that they were entering in 1954 and 1955. So I think that may have been their farsightful insights. But I haven't delved into exactly what was truly motivating. I'm kind of guessing right now.
Okay, thanks. Next question. Do you think that testing the ionizing radiations on fruit flies, as Muller did, could provide a good estimate of their effects on humans?
I think in qualitative terms, I think that it could. I think in quantitative terms, it probably would not be a particularly good idea. And that is that it's very difficult to extrapolate, in a quantitative sense from one species to another. There's a lot of uncertainty with that. It's very difficult in the world of toxicology to extrapolate even from a mouse model to a rat model, let alone from a mouse to a human model. There's a lot of uncertainty there. Even after Muller's work was done, and then there was research done by out of Oak Ridge with mice that showed that there was, I think, about a 15-fold difference between the mutation rate in the mice versus the fruit flies. But then, Muller's was just, one species of fruit flies. There are many ones you could have studied. There are the same for various mouse species. There are some that are very susceptible, some that are less susceptible, and so this is a very difficult area in terms of providing quantitative extrapolation. Qualitatively, I think that these models are very useful. They can tell if a mutation is occurring, if it's not occurring, and if it qualitatively happens, then it suggests that you should look more carefully at the species of interest.
What's your opinion on the reason why the regulations on radiations have been based upon a scientific fraud?
The regulations really are for the most part, come out of this recommendation that came out of the Baer 1 committee back in 1956, in which a fundamental decision was made to switch from threshold to linearity. And everything falls from that, followed from that. And essentially, what happened was that this was I'd have to say it was a political, philosophical decision that the radiation geneticist community had, and they believed, in my opinion, that they wanted to perhaps save the world from mutation, save future generations from mutation. They may have been, from what I can read, well-intentioned people. However, they basically gave up their scientific, they used their scientific credibility to make decisions based upon their philosophy. And what they owed the world, what they owed the country, they owed the world, was not their philosophy. They owed the world their scientific judgments. And then society and its political leaders could then weigh how the science fit into political judgments. But I believe that the radiation geneticist community in that 1956 committee report essentially gave up their, they had an appeal to authority. The authority was really in Muller and Stern. It's a very difficult situation to figure exactly, because when all those committee members went to that committee, they already had their minds made up. They were already believing in linearity, and I don't believe. See, this is an interesting thing, and that is that I've gone back and looked at the publication records in detail of every one of those members of that Baer 1 committee, and there were only about 3 or 4 that actually had significant experience with doing low-dose studies. Most of the other ones, they were men of achievement, but they were other kinds of achievement. And so they did not have experience with how to design and conduct low-dose studies and all the experimental nuances that you have to know and problems when you do those kinds of studies. So in effect, what they did was they had appealed to authority, to people like Muller and a couple of other ones who were on that committee who also had bought into Muller's views. And essentially, they allowed their decisions to be, they passed them through. They just appealed to authority. And Muller became, and just a few others, 2 or 3 others, became the key people that influenced policy for the rest of the 20th century. It all came back to the deceptions of the Stern studies. It's actually very amazing. It's hard to believe that it turns on a dime. But it's a very narrow point that it turns to. It reminds me, when the U.S. Challenger fell out of the sky back in the late 1980s. And it was all because several O-rings should have been replaced and didn't function properly. And all these very bright engineers in NASA, all these other talented people, everybody thought everybody else was doing their job. but actually somebody missed the O-rings, and people died and a whole disaster took place. Well, in this case, people didn't follow what was going on with Stern and Kasperi and Epoch and Mueller, and they appealed to authority. And what do we have? We have 60 or 70 years of an LNT regulation that was based upon what I think was fraud and deception, misleading, not providing all the information, substituting philosophy for science by people who were really outstanding scientists of the day, people that we looked to for guidance and that we trusted.
Okay, next question. Can you explain and give us an example on how a threshold value is determined?
Yes, a threshold. It's interesting that you raised this because it should be pretty easy. Basically, a threshold resonates within our common experience. For example, if people are listening to this, they would kind of have a sense of a threshold if you drink wine, and you might have a glass of wine, and you might not, or half a glass of wine, and you enjoy the taste, but you don't feel any sensory sensation, a slight lightheadedness or spinning in your brain, so to speak. But if you have 2 or 3 glasses of wine, you may begin to start to feel the psychological effects of the wine. But a fourth of a glass, half a glass, you may not. And so at some point, you pass a threshold and something happens. Below that level, there is no biological effect that is detectable. And so that's pretty much within the common person view what a threshold is. In a statistical sense, how we look at it is that if you get below the threshold, we expect there to be what we call variability or noise within a system, and you would expect random bounce below the threshold, but no significant deviation from the unexposed control group. So you can see, but both perspectives should agree with each other. The perspective of the common person and the glass of wine should not be any different than a biostatistician taking a look at random bounce below an estimated threshold. They're both telling you the same thing. There should be no significant biological effect or detectable biological effect below a threshold. It should be, from a radiation mutation point of view, it should be safe below the threshold.
By the way, are we in the presence of a threshold also in the hormetic model?
Well, the hormetic dose response is a biphasic dose response. In the case of mutation, the case of cancer, the case of radiation, we're looking at a J-shaped dose response, so that when the dose is out high, you see a dose-response relationship such that the dose goes up, mutations go up proportionately, and so too would a cancer risk. But as you titrate the dose down lower and lower and lower, according to the hormetic model, you reach a point which is really the equivalent of a threshold, in which there is no longer any treatment-related effects compared to the control. And you might think that, oh, gee, there's no effect as you lower the dose further. But actually, in the hormetic model, if you lower the dose further, we observe that the risk actually dips down below that of a control group, and therefore shows a J-shaped dose response compared to a straight line, then a flick up in a threshold response. The hormetic dose response is that which I spend a lot of time studying, and I find this to occur for essentially most chemicals and for physical stressor agents like ionizing radiation, and that it's independent of biological model endpoints. Biological organization occurs in the cell, the organ, and the organism, and occurs actually independent of biological mechanism as well. It's a very general phenomenon, and it's It's getting a lot more attention today in the pharmaceutical industry, in the chemical industry, and actually the ionizing radiation, non-ionizing radiation area as well. Not just from a regulatory point of view, but from therapeutic applications as well. And so how to make these new insights on the nature of the dose response more helpful to public health and for therapeutic application.
Why do you think that the majority of the general public wouldn't believe or doesn't believe in the existence of a threshold?
Well, I think that the way toxicology is communicated, dose response information is communicated with the general public. For example, if you were to take a look at all the chemicals that are regulated by the international community in their own countries, they're very similar. Let's say for drinking water contaminants. And you could look at non-carcinogens and carcinogens alike. And when you take a look at the number of molecules that it takes before you begin to see, these are even strong carcinogens, before you see responses taking place, you actually have to have an individual exposed on a daily basis for 70 years, more or less. People exposed to anywhere between, I'm going to just say, probably 10 billion to maybe 10 trillion molecules per day, every day, for 70 years for powerful carcinogens to begin to show a carcinogenic effect. And I'm talking, what that means over 70 years is that we're talking 10 to the 22nd or 3rd or 4th power of molecules, carcinogenic molecules, to your body to have been exposed to begin to show the beginnings of an effect. If one were actually to look at this, it would just show how actually incredibly unbelievable and lacking in credibility the LNT model has when it's applied to anything. It's how the risk communication message has been framed. It's been held captive by regulatory agencies whose mission in many ways has been to preserve their regulatory positions and jobs, to frighten the public into concerns that weren't justified. And I think that these are, I don't mind being challenged by those that I'm challenging. I would say, show me where my interpretation is incorrect. But I have gone back to all the regulatory estimates. I have done the calculations, and I have to admit I was surprised that actually I thought carcinogenic agents would be much more active at lower doses than non-carcinogenic agents, but actually it's about in the same realm of about per day, somewhere in the vicinity of probably 10 billion to maybe 10 quadrillion molecules per day before you see any change in the biology. That's actually pretty amazing when you take a look at humans and you say, well, when you hold up a glass of water, and a glass of water that has a contaminant at a drinking water standard, and that drinking, you might say that's perfectly safe, and that perfectly safe might have, in fact, 100 billion molecules of a toxic substances in it. You're looking at the glass looks nice and clear. You can't see the 100 billion molecules in it. You'll drink it, and you think that's nice and safe. And it probably is nice and safe, but in that, maybe 100 billion molecules of of a regulated toxic substance. And it tells me how toxic is it really if we're allowed to have at an approved drinking water standard by a regulatory agency 100 billion molecules in it. Yes, it needs to be probably regulated, but let's see this in relationship to the LNT, because the LNT is saying that there is no safe level and that a single molecule can initiate this pathological process, when in fact there would be 100 billion molecules, and you would need to have exposure to those 100 billion for your entire 70 or 80 year lifespan. I mean, you have to see that there's a discontinuity between what the regulatory policy is and what the actual scientific issues are and understandings are. They're really, they need to be addressed. And the LNT concept actually needs to be put to great scrutiny. It was adopted with no scrutiny. It was essentially pressed forward on the basis of both ignorance and fear and philosophy, and as far as I'm concerned, misrepresentation of the scientific record by leaders that we've talked about today.
Okay, next topic. Can you talk about the process that would enable the human body to protect itself against radiations by means of radiation preconditioning?
Radiation preconditioning. This is the concept of preconditioning itself is very significant. The radiation preconditioning is, in fact, a subset of that. But preconditioning, for the audience, is seen when a low dose of an agent, very low dose of an agent, is given prior to exposure to a very overwhelming or high dose. And you try to see, did that initial low dose given maybe a day or two before the high dose, did it affect the toxicity of the very large dose? And it could be a dose that kills an animal or makes it sick or does something like that. And actually, in many cases, the low prior dose can profoundly protect against the adverse effect by the more massive dose given subsequently. I know from work that we have done with a chemical called carbon tetrachloride that you could give a very, very low dose of carbon tetrachloride, one that doesn't cause almost any ostensible changes in the organism. One day later, you give a dose that will kill 95 to 100% of the animals, and none of the animals die. It protects them from that subsequent situation. You see that comparably happening with low dose of radiation. You see that happening actually. This is, for people who are listening to this, something that might be really relevant to them. Good portion of us are going to die from heart attacks or something related to heart attacks. I mean, 40 or 50% of deaths in the United States are due to heart-related conditions. Okay, researchers at Duke University in 1986, they provided a relatively modest hypoxic stress to dogs. And then a day or two later, they gave a massive myocardial infarction or a heart attack to these dogs. And they found that the dogs that got the mild hypoxic stress a day or two before had essentially 70 or 80% less damage than the dogs that didn't. It was, the investigators coined the term preconditioning. And then it was applied to many other systems so that you could actually protect the brain by preconditioning, you could protect the heart by preconditioning, the liver, the skin, the kidneys. And then after that, people also found that you could protect the body after you've been damaged by post-conditioning. And all these are now being implemented within medicine. How this relates to dose response, and that is that if you could take the preconditioning dose and you could give very, very low to much higher preconditioning dose, then follow with a large dose, you'd find that the dose response was an inverted U, just exactly like a hormetic dose response. So preconditioning is a manifestation of the hormetic dose response concept. And that's why I'm particularly interested in studying this phenomenon. So it relates to the world of radiation, radiation biology, radiation therapy. There's so many things that could happen in medicine that you could use a low dose of radiation prior to giving patients a massive dose of radiation. You could protect against subsequent damages. People now find ways to use preconditioning for patients before they have massive surgery so that they would enhance their recovery during the surgical process. This is like a wonderful new opportunities that are emerging on the healthcare system. And these are also, I can give you another example too, regulatory-wise, we published this from our lab, but US EPA and many other regulatory agencies, they'll say that if you take a kidney toxin and you take a second kidney toxin and you add them together, the response is additive. It's like two bad things, 1 + 1 = 2. Well, we took mercury, inorganic mercury, which is a kidney toxin. We took inorganic lead, which is a kidney toxin. We gave the lead 1 day before we gave the mercury. According to EPA, you should add these together because they should be additive. But because we gave it in a preconditioning sense 1 day before, then gave a strong dose of mercury, we reduced the mercury toxicity by 70 or 80%. It was just like what happened in the dog study. It really showed that the regulatory approach on chemical mixtures, for when you separated it by time within a conditioning framework, was basically not supported at all. And so it's a new toxicology today. And much of our toxicology was based upon ideas that need improvement. And essentially, it's very difficult, though, for regulatory agencies to change, to admit they made a mistake, and to follow the data. They're tied into defending past decisions, even when the past decisions can no longer be supported and are generally known as wrong, accepted as wrong. And that's the case really with LNT.
By the way, what quantities of lead and mercury are we talking about?
Well, these, I can't recall exactly those amounts because it's a number of years ago and the study was in mice. But this is a general phenomenon that would occur in the concept of preconditioning will occur. It can be very, very minor. For example, you could see, for example, in rodent studies, that you could actually take a blood pressure cuff. Okay, you take a blood pressure cuff, and all of us have had our blood pressure taken. People, tighten up your arm, block the blood, release it, and measure the return of your heart blood flow. Well, you could take a blood pressure cuff for a rodent and wrap it around its thigh, and there are protocols, and you could squeeze it a few times, tighten it up, let it go, tighten it up, let it go, and that's a preconditioning stress. Something as minor as that, and you could cause damage to the heart or the brain. That preconditioning stress, if you waited a day or so, would actually result in protecting the brain from damage. It's the same concept using the low-level lead against mercury. It's a stress, just like dietary stress. You could people have shown that if you have food and you take it every other day in an animal model, the act of stressing the animal so that it misses food for a day results in the upregulation of many different adaptive responses that then protects you from subsequent stressor agents that you could give the animal in its environment. What we're learning about today is a whole series of ways that the body has to protect itself against low-level, moderate-level, high-level stress by essentially using I'll call them preconditioning vehicles. These are mediated by exercise, maybe by other kind of dietary stresses, could be chemicals. Even low doses of radiation can serve as that type of preconditioning stress. And so it's very significant in what's happening and what will happen down the road.
Next question. When one talks about radiations, the thought of most people goes immediately to the accidents of Chernobyl first and Fukushima more recently. I read from your CV, page 24, that you participated as a guest editor for a publication with the title Distribution of Artificial Radionuclides in the Abandoned Cattle in the Evacuation Zone of the Fukushima Daiichi Nuclear Power Plant. On page 29 of your CV, you mention your 2011 paper, Improving the Scientific Foundations for Estimating Health Risks from the Fukushima Incident. Can you please talk about these studies?
These were evaluations of some of the scientific literature that might have relevance to assessing risk at Fukushima and perhaps other places as well. And the one thing that I think is the big message here, and that is that even though there can be improvements in the science and there needs to be improvement in the science, if you fit all your science through a linear dose response model, it's all going to come out, the interpretation is going to come out wrong. Because what you end up doing in any human study with populations, or you do this with animal models, you still have to somehow extrapolate down to levels that are extremely low. If you go from an occupational exposure or an animal exposure. And so what I have tried to do here in the papers and elsewhere is to try to say that our fundamental way of assessing risk was wrongly constructed, and it has led to incorrect estimates of risk that are applied to Fukushima, applied to Chernobyl, applied to other places. And as a result of those grotesque overestimates of risk, that they result in people and governments making policy decisions to shut down areas, to evacuate people, to have all kinds of actions take place that sometimes are far worse than the exposures to agents that we have. And so what we really need is a much more scientifically defensible risk assessment process that is consistent with the toxicological and epidemiologic literature, and basically what happened was that once you believe that a single ionization can initiate the process of carcinogenesis, there's no return, because everything then becomes fearful, and you think there's a risk with a single anything. And as I mentioned to you a few minutes ago, the amount of chemical carcinogen in drinking water, but you could relate this also to radionuclides as well. But we saw in EPA drinking water standards that you could be exposed to 100 billion molecules and more per day for an entire lifetime, 70 years, and have no increase in background in measurable cancer risk, none. And so the model is totally wrong. And yet if you were to apply an LNT model to this, you'd be concluding that in fact that a single molecule is a concern. And at least to the general public, no level is safe. And it's the wrong message. It's not the scientifically defensible message. It's a what is it? I call it a radicalized perversion of science that is now, like a disease, captured government policy when it comes to risk assessment. It's, in my opinion, it needs to be challenged and it needs to be changed. And this would play out, events would play out management very differently in places like Fukushima and other places. Because there's also a psychosocial component to this as well. And that is when you frighten people unnecessarily, then you change the health status of the whole family structure. And the mind can play strange games on the body, health-wise and social relationships-wise. This is a very serious situation, and it needs to be confronted. Yet if you challenge this, as I'm trying to challenge it, what happens is that people attack you for your motives. They make up motives that don't exist. And all I'm looking for is for people to look at the data, and let's go from there together and see what the data actually yield.
Is the hermetic model beginning to be considered by the nuclear regulatory commissions?
I think that the hermetic model is growing in its scientific acceptance and utility. It's widely used by the pharmaceutical industry. Now, essentially, for all those who are listening to this, essentially every single anti-anxiety drug, anti-seizure drug, memory drug, many others, they are based upon hormetic dose responses and the preclinical data with the mice and rats and whatever animals that they use. They have infiltrated and are totally pervasive in actuality in the pharmaceutical literature. Even those people who totally would be opposed to hormesis, they don't know this, but many of the drugs that they take, pharmaceuticals that they take to preserve their health, is based entirely upon a hormetic dose-response model. They're totally accepting it in one domain, not knowing that they are, and then in this other domain called environmental regulation, they say, oh, we can't go down this road, when in fact they've embraced it in their family and personal life. It's a very interesting contradiction. But I think that things have to actually change within regulatory agencies such as the US EPA, but others, in which they create the conditions of the game. In the game of risk assessment, EPA has actually defined risk assessment in such a way that it excludes the concept of benefit. Hormesis could actually incorporate a benefit into the equation. For example, if a low dose of radiation extended life, that would be significant to know, or some other chemical did, or reduce the occurrence of certain kinds of illnesses or adverse effects. That'd be very important to know. EPA would never consider, if an agent extended our life by 30 years, EPA would never consider it. They would regulate the agent outside of to be exposed, essentially, and to avoid the benefit. It's not a way to spend tax dollars' money, as far as I'm concerned. But the interesting thing along these lines, and that is that if hormesis were to show, and it could show this, that a low dose below the threshold were harmful, let's say a low dose of a drug enlarged a prostate gland, and it could do this in an inverted U biphasic dose response, EPA would say, okay, we'll set a standard based upon that. So hormesis is fine as long as it shows a harmful effect. But hormesis is not fine if it shows something that is potentially beneficial. That kind of dichotomy, inconsistency, makes no sense to me from a public policy point of view. It really needs to change. You have to follow the data and do what's best for the general public.
All right, last question, just an evaluation if possible. How would the safety cost of a nuclear power plant reduce after honestly reviewing the LNT?
Well, I think that there would be profound savings within the community. Put it this way: the industry, the payers, the users, right across the board. It would be, if LNT were replaced by a threshold model, hormetic model, you would have profound savings from top to bottom. And the interesting thing is that not only would you save tremendously in money, but the general health of the public would be significantly enhanced. It's difficult to understand how the public tolerates regulatory communities that impose costs on them and reduce their health status. But that's what our regulatory position has been within the U.S. and other countries, and it's my hope that in fact that rationality and following science will lead to a reversal of these kinds of actions and positions in the future.
Professor Calabrese, thanks very much for your time and for being with us.
Thank you very much for the opportunity.